Okay, good morning, guys.
Good morning, sir.
Good morning,
Good morning,
sir.
sir.
Good morning. There are only 12 of you, I think, but I guess many didn't know it was a working day. And yesterday was a holiday, and today is a working day. Today covers for Friday timetable. Okay. Any case, let's go on. So today we're going to continue from where we left off for polioviruses or picornaviruses. I told you what herd immunity was about in the last class. It's all about immunity of the community and protecting people who don't want to get vaccinated, who are not eligible to get vaccinated. So I don't know how many of you are aware. In the US, at least, there is a big group of people called the no-vaxxers, and they don't like their kids to be vaccinated. And we'll talk about it on another day in the context of another virus. So, but that is true. There are people who do not want their kids to get vaccinated. I'll tell you more about it in another class. Okay, today we're going to look at the lab diagnosis of poliovirus. So, if somebody has poliovirus infection, go ahead.
I have a question. Sir, is there some way that if viruses are affecting humans more than something? Is there ways some natural ways to trigger them and make them less effective so that they become less severe? We know that in oceans every day 20% of biomass is degraded by viruses, and if they don't degrade it, the environment will the ecosystem will not function in that way. Like, I was thinking that in case there happens some mutation in a virus, and instead of killing 20% of marine biomass every day, it is they start killing 30% or 40% of marine biomass daily. Then also the ecosystem will completely start dysfunctioning, and balance will the balance will not will not remain. So I was thinking, like, isn't it that in marine also the bacteriophages which are killing the bacteria get mutated at some point of time? If they get mutated, then what can trigger them so that the feedback so that in feedback they also are being killed, and somehow the environment remain ecosystem remain functioning well, and in that feedback system, what can trigger viruses to go down?
Actually, you have a long question. I'll give you a short answer because we have to move on. Your question is, what will happen if bacteriophages that kill marine life every day to maintain the ecosystem, if they start killing more or less? Okay, here's the short answer for that. The short answer is that bacteriophages have very high specificity. Even within a given organism, they can only infect. There are phage typing. There's something called phage typing. So if you take, let's say, this is, okay, I'm just going to draw 4 different bacteria of the same species. Okay, they have subtle variations, like, you call omicron, we all alpha, beta, so something like that in bacteria. I'm not going to go into the detail, but they are the same bacteria. A phage can perhaps, usually a phage infects only a very specific requirements it has. So it will infect only this bacteria. It will not infect this, or this. Although they're all the same bacteria, they have subtle differences. Even among the same bacteria, it can only infect one particular strain, or I won't get into that because that will trigger another debate. But it is a slight variation of a bacteria, subtype, let's say, of a bacteria. It will only infect that. So that keeps that restricts the range of what phages can infect, and that maintains the balance. Okay, I think I think that we'll stop there and we'll go on because I have to finish some of these things. But you can definitely call me and ask me if you have more detailed questions. You can also stop me with other questions, no problem. But for this, I think we'll just have to move on because I was supposed to finish this last class. Okay. So how do you diagnose poliovirus infection? If somebody has a poliovirus infection, how do you diagnose it? So anybody wants to take a guess as to what sample you might want to collect? Think of the pathogenesis, and that's going to help you.
Sir, stool sample.
That's correct. Stool is a good sample. What else? So you could also collect the throat
Sputum.
Mucus from the tonsils.
Yeah, throat swabs are swabs collected from the throat, just like they do for coronavirus, throat and the nose. This is the throat swab. It also collects feces. Very rarely CSF. I'll tell you what CSF is in a bit later in today's class. It's cerebrospinal fluid, the fluid that cushions the brain. I'll tell you more about it in a bit. Okay. What do you do with the swab? You put it, you try to put the swab into cell lines and see what happens, and usually they produce what is referred to as cytopathogenic effect. Now, I told you already, the cytopathogenic effect produced by each virus is usually specific for the virus. Now, if you add a throat swab from a poliovirus infected person into cell culture, this is what it will look like in 24 hours. We started here where I am drawing the arrow, and within 24 hours, you end up here, and it looks like this. If there is poliovirus, what do you see? We see characteristic cytopathogenic effect. It varies for virus to virus. You don't have to by heart it. Here you can see rounding of cells. You can see clumping of cells. You can see cells have broken off the monolayer. See, this is very dense. There is no gap. Here you see a lot of gaps. See, I'm navigating through the gaps. So breaking off the monolayer, rounding, clumping are some of the characteristic cytopathogenic effects of poliovirus. Now, if you see something like that, you suspect. Yeah, go ahead. Go ahead.
So, confirmation about polioviruses by visually looking at the through the microscope.
That is not confirmatory, but that is tentative diagnosis. I'm just coming to the confirmatory diagnosis. No, very good question. See, just looking at something like this, you cannot label somebody as having poliovirus. Then what do? You do something called neutralization tests. Now, what are these neutralization tests? They are very simple tests. I'll just draw a quick cartoon so that you can understand it. So let's say this is the poliovirus that has grown in cell culture. I'm just drawing very quickly. Give me a second. How many types of poliovirus are there? Anybody? While I'm drawing this?
Three.
Three.
Three
Types of polioviruses, right? Okay. Now, give me a second. Okay, I'll draw it like this. Okay. Okay. So I have in the laboratory antibodies against poliovirus 1, antibodies against poliovirus 2, and antibodies against poliovirus 3, the 3 types of poliovirus. Now, what I do is I got cytopathogenic effect that looks like poliovirus. If I take that supernatant, for sure it has some virus, most likely poliovirus, because of the way it looks, but I have to confirm it like one of you rightly said. How do we confirm that it is poliovirus? Now I take the supernatant from that cell culture, which has a lot of virus, whatever virus it is, you want to confirm it, you think it's poliovirus. Now, now assume that it has a triangle shaped epitope on the surface, and that's the on the virus surface. And you add poliovirus 1, 2, and 3 antibodies, and this is the matching one, poliovirus 2. Okay, you add it to the supernatant. You take the supernatant, you can concentrate the virus by some methods, don't worry about it. Then you add the antibodies to 1, 2, and 3 in 3 separate tubes. Virus plus poliovirus antibody 1, poliovirus 2 antibody, poliovirus 3 antibody. You don't know what the virus is. Then you re-inoculate this one, and this one into the same into cell culture again. Now assume that this triangle and this triangle are a match and they are neutralizing. this virus has bound and neutralized, whereas these two, 1 and 3, did not bind. They did not neutralize this. Now, if I inoculate this tube, and this tube into cell culture, which tube will have growth, or which tubes will have growth?
Third tube.
Third will have. One is also not neutralizing. Will it have growth?
Yes, one will also have growth.
1 and 3, which did not neutralize, will have growth, but poliovirus antibody 2 that I added to it neutralized the virus, which means it's a perfect match. And because it's a perfect match, it blocks the receptor, and this virus cannot reinfect the cells because the receptors are blocked. There won't be any cytopathogenic effect in this tube 2. Then you know this virus belonged to poliovirus type 2. Does that make sense to everybody?
Yes, sir.
Yes, sir.
Yes.
Okay, good. So that is called neutralization test. And that, like one of you rightly pointed out, you just looking at the cytopathogenic effect under a microscope cannot confirm a diagnosis. You need better tests for that. So this is one way of identifying it. What are some other ways of identifying it? You can also take the supernatant, which has now amplified the virus, and you can do what is referred to as an RT-PCR. So you can do an RT-PCR directly from the sample also. Now there are improved methods to detect even like 10 copies, 100 copies of the virus. You can detect it. But we did not have enough molecular power in countries such as ours, where we could not do much of RT-PCR. RT-PCRs were limited to metro cities or big cities. But now with COVID, I think it has changed the way of microbial diagnosis. There are going to be more and more and more RT-PCRs from this point forward, because India has increased the capacity to do RT-PCR like almost 100-fold during the last 2 years because of COVID. Now, you can take the throat swab and do a RT-PCR directly, and that will save you some of the time necessary for growing and other things. But growing it always increases the sensitivity. Even if you have fewer viruses, you can pick it up because you amplify the number of viruses before you do the RT-PCR. Now, RT-PCR also has other important applications. What are those? I told you we use the oral polio vaccine. Excuse me. And the oral polio vaccine is shed in the feces. It can infect other people. So if somebody has poliomyelitis, very rarely it can be vaccine induced, right? 1 in 3 million or something of that order. So that can be vaccine induced. You can actually check if the sequence of the virus belongs to the wild type or the vaccine induced type, because the live attenuated vaccine contains mutations that make it not grow in the neurons. So you can actually differentiate vaccine sequences or the attenuated virus from the wild type virus by using PCR followed by sequencing. I'll try to explain that a little bit more in one of the coming slides. What about serology? When we talk about serology in microbial diagnosis, serology usually refers to antigen antibody reactions, but most often we refer when we say serology, we speak about identifying antibodies in a person's body. Now, why have I written this? It's not very useful. Why is looking at poliovirus antibodies not very useful for diagnosis of active poliovirus infection? Why is that? Guys, nobody wants to guess. Do you have antibodies to poliovirus if I look at your blood?
Yes, sir, because a vaccinated person can also have antibodies.
Pretty much the entire world is vaccinated, and pretty much
Yes.
all of us have antibodies. So looking at antibodies to poliovirus does not help you identify whether you are having an active infection with poliovirus or not. So that is not very useful. Antibody tests are not very useful because of that reason. I already looked at the slide. I told you what it is. Now, this is just an example slide. You don't have to, again, by heart anything here. This is just uninfected cell line. in the same cell line, adenovirus produces CPE like this, herpes virus produces CPE like this. So the only reason I have this slide in here is to help you conceive the idea that cytopathogenic effects or CPE induced by each virus on the same cell line is remarkably different, that allows a tentative identification of the virus. Although it may not be specific, you might need more tests, but it allows a tentative identification of the virus. That's the only purpose of this slide. Now,
Sir, do we have specific cell line for polioviruses?
yes, so there are, we usually, I mean, poliovirus can grow on multiple cell lines in the laboratory. Classically, they used to use something called primary monkey kidney cells, which used to support poliovirus growth very well. Now they use a variety of other cell lines also. So I won't say there are, poliovirus can grow only in one cell line. See, most diagnostic virology labs will have, let's say, about half a dozen to a dozen cell lines, and they will help diagnose most human infections that we encounter. And what has happened in the last 20 years is that we are slowly moving away from cell line based diagnosis, because everything can be amplified by a PCR or a RT-PCR and then detected much faster. But the answer to your question is yes. Usually there are 2 or 3 cell lines that are used for growing polioviruses in the laboratory. And if you suspect a particular group of viruses, then you have to use the appropriate cell line for diagnosis. If the clinician will write on the request throat swab, query poliovirus or enterovirus or a query paralysis, something like that. And based on that, the cell line to be used is chosen by the virologist. Is that clear?
Yes, sir. Yes, sir.
Okay. Now, I'm going to tell you something very fundamental that most of you know. I'm not going to play these, but you can watch it if you are not really familiar with PCR. I just have a very simple cartoon here on PCR. Can one of you volunteer to tell the class, explain the 3 steps? So this is step 1 here. What is okay, step 1, step 2, and step 3 of PCR. Can anybody try and explain this in very simple language to others in the class? Because there are some people who may not be very familiar with PCR. Very quickly. Nobody?
So, can I?
Yeah, go ahead. Anybody? One of you.
Uh,
I am on full
so,
screen mode. I can't see who is talking, but it says PT here. Go ahead.
So in PCR, what we check, we amplify the certain DNA sequence with the use of primers that are having complementary to uh, 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 starting and the end of the DNA sequence. So what polymerase do? Polymerase extend these primers and amplify the our template DNA that we have in original, and amplify to several million, thousand folds or million folds. So in denaturation step, what happens at the higher temperature, the double-stranded DNA will get denaturated and it becomes single-stranded. And in annealing temperature, when we have to anneal our primers to the single-stranded DNAs, then we have to lower the temperature. So say 55 degrees, 50 to 70 degrees Celsius. So after annealing, the polymerase will come and will function at its what we say optimum temperature, which is 72 degrees Celsius for tech polymerase we generally use, and this will extend the single stranded to double stranded form, and the cycle continues in again then after setting temperature to 95 and then 55 and 72.
Yeah, if there's anybody who did not understand this very well, I can try to go through it very quickly. If everybody is good, we can move on. Is there anybody who has a question about PCR? I think most of us know about PCR because of SARS-CoV-2. Everybody understands, right? You started with 1 copy of double-stranded DNA. Now what you end up with is 2 copies of double-stranded DNA at the end of PCR, and it's just like Xeroxing, right? So it just copies wherever you put your primers. That marks the boundaries of what's going to be copied. and this is copied every cycle, theoretically it doubles the number of copies you started with. Let's say you started with just 2 copies or something of that order. In 36 cycles, you will end up with this many copies, and this can happen in 1 to 2 hours. And so that is what is shown here, that you can amplify exponentially the number of virus genome material that you have using PCR. And how do you detect after PCR? Typically, you this is also changed now. I won't talk about it now, but still many labs do use this. You run something called an agarose gel electrophoresis, and when you do that, let's say this is a molecular weight marker, 100 bases, 200 bases, 300 bases. Let's say these 3 samples, which are, let's say, roughly 340 bases, just an example. Okay, so if you have a specific size, you know the specific size of your product because you designed this one and this one, the 2 primers, you design them. So you know where the primer binds, that's going to be amplified. And if the distance between the 2 primers is 340 bases, and you get a 340 base amplicon, that means that sample contained poliovirus, this sample, and this sample. And for example, if you ran something here and it did not have a band here, that would be negative for poliovirus. So this is how it is detected. Now, I told you that PCR can also be used for identifying the wild type strain and vaccine strain. Let's just say, I'm just going to draw something crude here. Okay, so maybe I'll just draw it here. Okay, so let's say this is the wild type strain and this is the vaccine strain. And this in the 340 base region, this contains, let's say, 3 mutations. 1, 2, and 3 mutations. This is because the attenuation is done by mutations, right, which are induced by growing it in the wrong cell line. So if you amplify the wild type strain or the vaccine strain, you still get a 340 base product because the mutations are not where the primer binds, they are somewhere in between. Then if you send these 2 products for sequencing, let's say this is the vaccine's wild type strain and this one is the vaccine strain. They both give you the product of the same size, but if you sequence them, you will identify 1, 2, 3 mutations in the vaccine strain, and that will help you nail that this is the vaccine strain and this one is the wild type strain. You can distinguish them based on the sequence. You can amplify the product, sequence it, you'll know whether it's wild type or mutant. Now, moving on to other picorna viruses.
Sir, I have a question.
Go ahead.
Sir, is it mandatory to give samples for sequencing, like if
No,
it is vaccinated or
no,
every
no,
time?
it's not. But sometimes actually, the clinician will say that the child got vaccinated 10 days ago and has reported with poliomyelitis. Then you want to
Okay.
distinguish whether it's that 1 in 3 million cases, which is vaccine-induced poliomyelitis, or the child actually got infected with the wild type. That is one reason where you'll have to do it. But now in India, if you have a poliovirus case, we haven't had a case in almost a decade now. We haven't had,
Yes, sir.
so we are good. But if you have a case in India that presents with polio-like symptoms, definitely they will sequence it. The reason is actually because it has not been there. If we have a case now, it's going to be most likely vaccine induced only. You understand, right? What I'm saying?
Okay, sir. Yes, sir. Yes, sir. Thank you.
So, yeah.
Sir,
Yeah, go ahead.
can ELISA be done or used as a diagnostic tool?
ELISAs are primarily good for 2 things. They can either detect antigens or antibodies. So if you have, if you're at this stage where you have this kind of CPE, one can actually develop ELISAs to actually test whether this has poliovirus 1, 2, or 3 antigens. That is a very easily doable thing. But the flip side of ELISAs is that they need, let's say you have only 2 samples for poliovirus. You still have to use almost 15 wells or 2 strips in an ELISA because you'll need a negative control. You need a blank then you need 2 negative controls, then you need high positive, then you need a low positive well, then you need perhaps sometimes you need some other standards in ELISAs. Then you test your sample in duplicate. So that makes it almost like for 1 or 2 samples also you'll have to use 10 wells. And because poliovirus, you don't get these samples on a day-to-day basis in any laboratory. ELISA is more difficult and PCR is much more easier, because for PCR, you're all set up. You just need to take that supernatant from this well right here, take the supernatant, extract RNA and do an RT-PCR. That takes only 1.5 hours. You have the result. So that is easier, although theoretically ELISAs can be done. But where are ELISAs used more often? In diagnostic virology, they are used for samples that you encounter in truckloads on a day-to-day basis. That includes, for example, screening for HIV. Anybody who undergoes surgery in a hospital, in a big hospital, there would be like dozens of surgeries every day, day out, day in, day out. There would be several dozens of surgeries. So pretty much all of those samples will come for screening of HIV, HBV, and HCV, which are blood-borne viruses. Why do they have to screen for it? Because the surgeon will be at risk if he's operating on these individuals. He will take extra layers of protection if they are positive for any of these. So these samples, for example, in big hospitals, just the screening samples come, or if they undergo some specific procedure, even small procedures in the operation theater. Like, for example, if somebody has like something like a boil and the surgeon has to just cut it and take out the pus, even then they do these things. And these are done in large numbers. For example, per day, the number of tests will be something like in a big hospital, it will be at least 400 to 500 tests like this every day. So then actually it's easier to do ELISAs than PCR because they are more economical. You see what I'm saying?
Yes, sir. Got it. Thank you.
Okay. Okay, where are we? We are here. Okay. We're going to move on to other picorna viruses. We are not going to look at any of them in great detail. I'll just tell you a little bit about each. So like snippets so that you can remember some things about other picorna viruses that will help us complete what are picorna viruses. So next to poliovirus, the most important group of picorna viruses medically is Coxsackie viruses. And like I told you earlier, Coxsackie is the name of a suburb somewhere in the US, in the East Coast. And that's where that's where this virus was isolated first, and it was named after the suburb. Now, it has pathogenesis similar to polioviruses, but they don't easily grow in cell culture. Most Coxsackie viruses do not grow in cell culture, unlike polioviruses. Then how do you grow them? You grow them using something called suckling mice. I'll tell you a little bit more about suckling mice in just a second. So unlike polioviruses, where you have only 3 types, Coxsackie virus type A, there are 24 types, and B, 6 types. Together, there are 30 Coxsackie viruses that are medically important. Okay, now almost none of them grow easily in cell culture. Give me a second, I'm so sorry. Okay, sorry about that. Okay, so there are 30 different types of Coxsackie viruses. They don't grow well. Almost none of them grow well in cell culture. You grow them in something called suckling mice. Now, what are suckling mice? I think my screen is frozen. Yeah, here we go. For those of you who have not seen suckling mice, this is the size of suckling mice. This is a palm, and this is the size of suckling mice. What qualifies as suckling mice? Mice that are less than 3 days of age. less than 3 days of age, within the first 72 hours of their birth, they're called suckling mice. And why is that important? Because like after 3 days, they get antibodies from the mother because and their immune system also fights back better. So that does not allow good growth of Coxsackie viruses. So you have to inoculate mice that are less than 3 days of age. And these things are slowly, people are moving away from this, although these are part of classical virology labs. Most big virology labs will have a stock of suckling mice. And if they have something, yeah, go ahead. Somebody had a question? No? Okay. So yeah, go
Actually,
ahead. Yeah, go ahead.
can't we use like skid mouse or neural mouse like immunocompetent because they also have like immunity system is not much more developed in that case?
That's correct. We can use, but they're much more expensive. But this can be any mice. That has to be a very expensive mice.
Okay.
Right? So this is much easier, and this is basically you just have a colony uh, growing in the virology lab, and you can just pick up anything that any mice that's born within the last 72 hours and inoculate, and you'll have a lot of these available easily at a very low cost. Okay, so then you inoculate the material from the throat swab or the feces. You do some processing and then you inoculate it into the mice, and if it develops paralysis or if it dies within the next 5-6 days, then you know that the sample had Coxsackie viruses, and then you can actually take that mice and you can take brain tissue or other tissue and do RT-PCR to confirm it, and that's how Coxsackie viruses are classically diagnosed. Now what is happening is you have better RT-PCRs that are able to directly detect Coxsackie viruses from the sample, for example, throat swabs. So you can do a PCR of a throat swab, but can anybody see a flip side, or what is the difficulty of doing it? Because you have so many types of Coxsackie viruses, almost 30 different types. It's difficult. Each one of them, you'll have to identify a very conserved region across these and then do a PCR. So that is the flip side. But when a mice is inoculated and it dies, again, they also do something called mouse neutralization test. So they have antibodies against all the 30 types of Coxsackie viruses. They take the virus from the mice that died, they purify it, they add it to, in 30 different tubes, they add it to antibody 1, 2, 3, 4, and then they inoculate mice again, and then they see which one is growing, which one is not growing. Ideally, only one will not grow, and the one that does not affect the mice, that's going to be the type of Coxsackie viruses. So this was the complicated way in which Coxsackie viruses were diagnosed, and for this precise reason, for example, in countries like such as ours, we don't have almost any data on Coxsackie virus diagnosis, because only very big diagnostic laboratories, for example, AIMS or CMC Vellore, or only 3 or 4 centers in India can diagnose these viruses. Now, going on to the next group of viruses, which are called echo viruses. Like I told you, echo stands for enteric because it enters and infects the GI tract. Cytopathic because it can grow in human cell lines and cause cytopathic effects. Go ahead.
So is the technique still the same for putting setting up 30 test tubes and identifying which Coxsackie serotype is it?
Yeah, and there have been some advancements. So instead of putting it back into the mice, they got something called LBM pool typing, which is again just adding the virus and trying to see whether it agglutinates or neutralizes the virus on a slide. That that you don't have to go back to the mice again. You don't need 30 mice again to inoculate. So that is one advancement called LBM pool typing, but you still need the 30 antibodies. The other way is, because you don't have all 30 Coxsackie types circulating in a country, big labs know actually 3 or 4 predominant types that come through the year. You don't have all the types. So what they can do is they can do a PCR for those 3 types. Let's say Coxsackie A16 and Coxsackie B4 are the predominant types in India or in a particular region. That lab can only test for those 2 PCRs. That is a possibility, and that is also being followed. Does that answer your question?
Yes, sir.
Okay. Okay, so these are echoviruses are they infect humans, they infect the GI tract, they can grow in cell culture and cause cytopathic effect. But at the time of discovery, there were no diseases associated with it. And there are 34 different types or 32 different types of echoviruses right now. 2 were reclassified. Don't worry about these numbers, but approximately over 30 types of echoviruses, right? But echoviruses, although they were named orphan viruses because they were not associated with disease at the time of discovery, today's world, they can cause a variety of diseases. So these 3 viruses are again picornaviruses. Among picornaviruses, if you recall, they belong to a group called enteroviruses because they enter through the GI tract and infect there, and then they move on to other parts of the body. They're called enteroviruses. So these 3 viruses, like there are 24 of these, 6 of these, and over 30 of these, right? So there are many of these viruses, and many of them can cause disease, including echoviruses, although their naming is now a misnomer, but because they were named, we stick to it. We have not changed their names, but it definitely does cause disease in humans today. I mean, we know this for several decades, but at the time of naming, it did not cause. That's it. Now, what kind of diseases do these viruses cause? They can cause what is paralytic disease. All of you know that is just like poliovirus disease. Meningitis and encephalitis. I'll tell you a little bit more about these 2 terms in a bit. I think I have a cartoon. Myocarditis. What is, anybody wants to guess what that is? What is the myocardium?
Inflammation of myofibrils.
Say that again.
Inflammation of cardiac tissue.
That's correct. See,
Inflammation of cardiac tissue.
that's correct. So anything that ends with itis means inflammation. So myocardium means the muscle of the heart. So inflammation of the muscle of the heart is myocarditis. Exanthems, for those of you who are not familiar, what does exanthem means? Exanthem means fever with rash. Like chicken pox is also an exanthem. Like I was telling you, one of you who said that they had chicken pox after vaccination, it's possibly, if it's not lab diagnosed, it could be false. So you can see that all of these viruses, almost 50 of them, many of them, in fact, can cause something called exanthems, which is nothing but fever with rash, which is the same thing as chicken pox. Sometimes it's hard to do a differential diagnosis unless you're experienced. Any rash does not mean chicken pox. So many of the enteroviruses can also cause that. Hemorrhagic conjunctivitis, I'll show you some cartoons and explain what that is. That can be caused by some Coxsackie A types. They can cause respiratory infections. They can cause what is referred to as PUOs, or it's called pyrexia of unknown origins. So when you have fever for more than 3-4 days and it's not explained, you don't have any other symptom, you don't have a running nose, you don't have diarrhea, then the doctor thinks about some specific diagnosis like malaria, typhoid, and a few things like that. And enteroviruses are also part of the differential diagnosis. They can also cause fever of unknown origin. And this one also, I'm going to explain it. Some Coxsackie viruses have been associated with pancreatitis or type 1 diabetes. I mean, this association is kind of not very conclusive, but there are more and more studies that are showing association. I'll talk about it a little bit more in the next 10 minutes or so. Okay, the receptor for many of these viruses is not known because it's difficult to identify a virus receptor. And they clearly, you can see that they can infect the brain, they can infect the heart, they can infect the skin, they can infect the eye, they can infect the respiratory tract. They don't actually infect these organs. I'll tell you a little bit more about the diabetes part. in a little bit. They can infect a variety of tissues. They must have something that is on the cell surface of many cells, common to many cells. That's why they're able to do this. Okay, what about enteroviruses and type 1 diabetes? Like I told you, some Coxsackie viruses, including some A and some B, although A is written as negative here, don't worry about it. Some Coxsackie viruses have been associated with type 1 diabetes. Now, what do scientists predict is happening there? Let's say I'm just doing this. This is a surface epitope. You have, when you get infected with a Coxsackie virus, your body will produce antibodies against surface epitopes of the virus. And accidentally, if you look at the islets of Langerhans, which are the cells that secrete insulin, they also have some similarity to this epitope. So these antibodies that are produced in the body in response to specific Coxsackie viruses are able to go and bind to the same person's islet cells, and they are causing some damage to the islets of Langerhans and the pancreas, which are the cells that secrete insulin, which is important for glucose metabolism, and which is associated lack of insulin leads to type 1 diabetes mellitus. Now, what are these antibodies going to be called now? They're going to be called, anybody? Autoantibodies, because
Autoimmune antibodies.
autoimmune antibodies, autoantibodies, they're going to target your own cells. Although your body made these antibodies against a virus, there is some similarity between some epitopes on the islets of Langerhans and the pancreas and this virus. These antibodies are able to cross-react and they're able to bind to these cells. This doesn't happen in everybody who gets this infection, only in some people. There are going to be additional factors which are responsible for this. But then these people may end up with getting type 1 diabetes mellitus. So this is a mechanism. These things were known only a couple of years ago. You can see that these papers were published only about 3 years, 4 years ago. And this is also the same thing. Now, there are also big pharma companies which are trying to make a vaccine. Go
Hello,
ahead.
sir.
Go ahead.
Actually, there is a term clonal selection is there. So in that case, clonal selection is not happening or any like problem occurring there so that they do cross react.
No. See, this is actually this is not just unique to this. So these things happen commonly in microbial infection. For example, there are some streptococcus infections. If you have those infections, antibodies against streptococcus, some of them actually go and bind to the valves in the heart and cause what is referred to as rheumatic heart disease. It has nothing to do with clonal selection, but it's not just the organism itself that's enough. There is some additional factor that we don't know, but along with the organism and some additional factors in some individuals, they're able to elicit antibodies that are able to go and bind to other parts of their body. In this case, islets of Langerhans, and then in streptococcus, they can bind to the valve in your hearts, the valves that are very important in your heart. They can go and damage those valves. So these happen by accident. They have nothing to do with clonal selection. What is clonal selection typically in antibodies? And we all have a repertoire of antibodies, which means we can make like 10 power 7 or something in that order of different types of antibodies. When we get exposed to a pathogen, that particular type of T cell and B cell that are able to recognize the pathogen, B cells only produce antibodies, so they proliferate more and they produce a lot of antibodies against that particular pathogen when we are exposed to the pathogen. So that is the clonal selection, but that has nothing to do with, see, autoimmunity and clonal selection, they don't necessarily meet. See, when we have an immunogen in the body, those particular T cells and B cells are activated, they do their job. They are produced in large numbers. They produce large number of antibodies. They become memory cells. All these things are done. That has nothing to do with autoimmunity. Is an accident in this case. Is that clear?
Yes, yes.
Okay. So we have only about 10 minutes. We're going to finish quickly the remaining slides so that we finish this today itself. So some companies are trying to make a vaccine, but those are not really important, just so that you understand the kind of research that's going on in this area. Now, what is this disease likely to be called? You have the hand, you have the foot, you have the mouth. What is this disease going to be called? You heard of this hand, foot, and mouth disease? Guys?
No, sir.
None of you heard about this? Delhi had a big outbreak in 2014, 2015. Millions of children were affected. School-going children were affected. Yeah, go ahead.
It looks like something measles, is it?
No, it's not measles. That's why measles is an exanthem. This is an exanthem. Chickenpox is an exanthem. But you need more expertise to distinguish each of them. The kind of rash that happens and the place at which the rash happens are the two key factors for a differential diagnosis based on symptomatology or presentation. But sometimes people misdiagnose based looking at this, but this is not measles. We'll talk about measles. You'll understand how this is different and why this is different. Let's just move on. This is called hand, foot, and mouth disease caused by Coxsackie viruses. There have been big outbreaks of hand, foot, and mouth disease in China and India in the last 7-8 years. They've been very big, sizable outbreaks. Millions of children were affected, including in Delhi particularly. And that is a few years ago. But whenever I do this class, I just Googled it, I think last night, and there is an outbreak that's ongoing in Jamaica. Few months ago, there was an outbreak in Brazil. So these are the outbreaks with Coxsackie viruses keep happening in different parts of the world. That's all. That is the take home. Now, I told you enteroviruses include polioviruses, Coxsackie viruses, echoviruses, and now any new enterovirus that is not poliovirus is being classified as new enteroviruses, and they're going to be sequentially numbered after EV78. And I told you I'll tell you a little bit more about acute hemorrhagic conjunctivitis. All of you know that the conjunctiva is other than the eyeball, which is black, the rest of the part is covered with conjunctiva, something like a mucous membrane that protects the eye. Now, if that gets inflamed, that's called conjunctivitis. And because it gets inflamed and the blood vessels become more dilated and the eye becomes red, it's called acute. It happens very quickly, acute hemorrhagic, because everything looks red. Inflammation of the conjunctiva. So this is caused by, again, a picorna virus called EV70. There were very big outbreaks in the 70s and 80s. Now we don't have much of them, but we do have them in pockets. Now, EV71 is one of the very important enteroviruses. It can cause poliovirus-like disease. It can cause pretty much anything that has to do with the brain. So it can cause what is referred to as meningitis. I told you what meningitis is. There are 3 layers of meninges that protect the brain. And there is something called cerebrospinal fluid that acts like a cushion in between the meninges, again, to protect the brain. So inflammation of these 3 protective layers, this is the meninges. That's called meningitis. It can be caused by EV71. And inflammation of the entire brain itself, that's called encephalitis. And EV71 can also cause that, but it may or may not be life-threatening, but there may be residual paralysis, just like with polio, which is lifelong. But a lot of times our body can fight this and get rid of it, but there may be residual paralysis that may be remaining. So this is EV71, because these viruses, we don't know the receptor. They can affect different parts of the body. EV71 appears to have a predilection towards the brain and the meninges. Because it's the brain, we know more about this virus. This is just for information's sake. EV72, originally it was called EV72. Now it's reclassified as hepatitis A virus. Don't have to know more about it. There are always like EV71 outbreaks happened in Colorado. Like I think this is last year, and this is again CDC has warned about a poliovirus like outbreak last year. So this is all just to let you know that these things keep happening. Now, how do you diagnose enteroviruses in the laboratory? Pretty much the same way as polioviruses. Throat swabs or stool or CSF. They are grown in cell lines or suckling mice for Coxsackie viruses, or you can use RT-PCRs pretty much the same way. Of course, serology is not useful because you have like 60 different types of enteroviruses, 60, 70 types of enteroviruses. You will not be, you'll not be able to identify a specific virus based on the presence of an antibody because you need that many different antibody assays. For one sample, you need to test 70 different assays. So that's not possible. They don't share antigens, so it's not easily possible to distinguish. Like let's say I can just say you have Coxsackie virus. Even that is not possible because they don't share antigens. They are so different from each other. Now, what do we do? Are there antivirals for these? No, there are no antivirals for enteroviruses, any of them. Okay, so what do we do with them? Usually only polio is preventable by a vaccine. Others, you don't have a vaccine because there are 70, 80 different types of enteroviruses. You can't have that many different viruses. It is not economical to have it. So you don't have a vaccine for others. But you do have something called human normal immunoglobulin, which is nothing but a concentrated immunoglobulin from blood donors, multiple blood donors. Let's say 100 donors you take immunoglobulins, pool them all, concentrate it, and take a small quantity, aliquots of it. That's called human normal immunoglobulin. What is it used to prevent? It's used to prevent congenital infections with enterovirus. What does that mean? If a mother has enterovirus infection, let's say Coxsackie, echo, whatever be it, and if you want to stop the child from having it, what you do is you can also give the mother this, you can also give the child this, and then the hope is like this, because there are so many different types, you don't know, you don't have a specific diagnosis. Even if you have specific diagnosis, you don't have specific antibodies. So instead of that, you just used pooled antibodies. You just have a cocktail of antibodies. See, I might have been exposed to, let's say, 3 Coxsackie viruses and 2 enteroviruses sometime in my life. It called mild disease, I don't know, but I have antibodies. You may have been exposed to a different one, third person, another one. So if you pool these antibodies, you have a cocktail covering pretty much everything or most of the common things. And that can prevent mother to child transmission of infection. If you give it to the mother, especially if you have to give it to the child also, it neutralizes the virus that the child gets from the mother, because you assume that the cocktail has antibodies to the specific virus infection that the mother has, and it has good efficacy or reasonable efficacy in preventing the child getting infected. So that's this. Last 2 minutes only I'll take very quickly. All of you know about rhinoviruses. They are viruses that infect, cause running nose. There's a commonest virus infection. Almost 2/3 of all virus infection are related to rhinoviruses. There are 200 different types of rhinoviruses, over 200 types. And that's why you keep getting them back again and again, because today you had a mild virus infection that caused running nose. Tomorrow, next, after 3 months, 6 months, you can have another infection. That's because they don't share sequence identity. They are so different from each other. One does not protect against another. So you keep on getting running nose because of rhinoviruses. And they have the ability to grow at a slightly lower temperature. Your nose and the sinuses are at a slightly lower temperature than your lower respiratory tract. They grow better there, and that's why they cause only running nose and nothing more than that. So they cause what is referred to as upper respiratory tract infection, and they can cause sinusitis. These parts of the our face are hollow and they're called sinuses. They make the head lighter and help reverberation, or they give a tone to your voice. Otherwise, our voice might be sounding very deep if you don't have these hollow bones. They infect these regions called sinuses. It's all in the upper respiratory tract. You can get it from aerosol from an infected person. You can get it by touching inanimate objects which are contaminated by an infected person. There is no need for diagnosis or treatment. It's a very mild thing. If you had rhinovirus infection, if you had a running nose because of a viral infection, what do? Just wash your hands and cover your cough. These are the two most important precautions. I think I've kind of overshot with time. Just about 1 minute left. Anybody has a quick question? No questions? All good? You guys are with me? You're able to follow
Yes,
this?
yes,
Yes,
yes.
sir.
Okay,
Yes,
okay then. Thank you guys. I'll send you the slides.
Thank you, sir.
Thank you,
thank
sir.
you, sir.
Bye bye.
Thank you, sir.
Bye guys.